Welcome everyone to this seminar, which is run by UNSW, but it's part of the Statistic Across Campuses initiative. So today we have James Thorson, who's a senior scientist and program leader of Habitat and Ecosystem Process Research. This is based at the Alaska Fisheries Science Center, which is within the National Maritime Fisheries Service. Jim has been spending a bit of time with David Wharton and his group, and he also agreed to give a talk today, so thanks, Jim, for that. And his talk is on forecasting non-local climate impacts from mobile marine species using extensions to empirical orthogonal function analysis. Jim, the screen is yours.
Yeah, thank you. Before I go on, thank you, Boris, for helping to organize this. Thanks to Dave for inviting me. It's been a fun visit so far. I can't say that I know the other talks that are given, and so this is sort of the themes. I think are kind of a perfect mix of you know, some ecological theory and then some technical statistical concepts. So it's kind of perfectly suited to, you know, miss everybody at some point, I'm sure. But I guess that the overall theme is trying to think about how we can do habitat science under climate change. So, you know, I guess I start here with this picture of a, you know, of a mobile animal and its landscape, and in areas like this in Alaska, you know, there's a lot of development that is being proposed and discussed and debated. You know, so for instance, there's a large mine called Pebble Mine that's being discussed in the Bristol Bay River system, and this and other cases are examples of sort of previously unknown, you know, unobserved changes in habitat that can potentially affect downstream conditions for marine animals. And so trying to figure out how we can deal with non-local effects on habitat is the general motivator for this. And I guess I'll do this talk in 4 parts. First, I'll set up what I'm going to be calling kind of the grand habitat challenge, the sort of issues for dealing with habitat science that I and colleagues at NIMS hopefully will tackle in the next 10 years. Next, I'll talk about empirical orthogonal functions, or EOF analysis. I'll introduce a concept called EOF regression. And then finally, I will tie it together, hopefully by trying to do ecosystem forecasting, so decadal scale forecasting of community reorganization using EOF analysis. And I guess, you know, this talk is an opportunity for me to think about and continue to refine ideas and also get feedback. And so one project I've been thinking about is, you know, how we can coordinate our different habitat science within the National Marine Fisheries Service. That's the 6 science centers in the United States that do science to inform ocean fisheries. And so on the left-hand side, I've got sort of this little directed acyclic graph representing, you know, or like a graphical model or a structural model representing how a change in biomass, a delta B in the top, would be affected by current biomass B, but also by fishery catch C, and some set of exogenous drivers X. And so, you know, we've got a lot of population models that try to predict density dependence to incorporate environmental information and deal with sort of known exogenous drivers like catch and how that affects population dynamics. And you know, of course, population dynamicists have been doing models like this for, you know, 100 years. So more recently, there's been an effort to do this, you know, as a habitat model, to define a population model for each location, each site that's defined spatially. And so on the right is sort of a toy model of how people might extend this to a habitat model. So you've got a change in biomass is the response. It's predicted by current biomass at each location S. And again, there's sort of a separate catch or impact at every location and a set of covariates X. And that's what I'm going to call this biomass dynamics habitat model. However, you know, I and others have been doing research about those sort of you know, population dynamics models that are defined for each cell, each raster cell in a landscape, or each site within a metapopulation. But I don't think it's very biologically plausible for several reasons. One of them is stage structured effects. You know, so population dynamicists for a long time have thought about stage structure. Partly it's due to non-local effects, so individual movement or environmental teleconnections will cause different locations to be correlated. There's also, you know, attribution questions dealing with direct and indirect pathways, and then dealing with counterfactuals or kind of causal analysis. You know, essentially estimating, you know, specifying a model structure where we can learn about what would happen under some previously unobserved, you know, human development or novel climate. And in the remainder of the talk, I'm going to talk about sort of non-local effects and specifically environmental. It doesn't seem to be advancing. There we go. So non-local effects. On the left-hand side is an extension of that model in which it connects every node of that graphical model with every other node. And so in that case, we've got all these red arrows and blue arrows and green arrows, and each of those, you know, could be ecologically plausible under some kind of circumstance. You know, so for instance, oftentimes exogenous drivers, you know, habitat conditions at an adjacent site affect population dynamics at a site, you know, that like some species prefer to be in an ecotone, or, you know, fishes prefer to be in an area with heterogeneous bathymetry. So dealing with non-local covariate effects is important. You can also have non-local density dependence. And so you end up with this oversaturated model that's not identifiable using the amounts of data we typically have. And I guess I'll advocate that, you know, we need to figure out how we can prune that set of arrows down to a set of arrows that's manageable. And in terms of density dependence, I think we can do that by better specifying advective diffusive dynamics within spatial models. And for covariates, which again will be the main theme of what I'm going to talk about, we can do this feature identification. One way of doing that is using empirical orthogonal function analysis, and I'll define that in a bit. But it's essentially a way of trying to automate sort of, given all of the different dynamics that we observe in nature, what are the major modes of variability that we want to pay attention to when we're trying to relate dynamics, community dynamics, to the environment. I'll just say that throughout this, I'll be using software that I have online. So if you go to fishstats.org, you'll get a splash page. And that includes links to the software FAST, standing for Vector Autoregressive Spatio-temporal Model. It's an R package that I use for you know, operational software that has, you know, version control and integrated testing and is fairly stable. There's also a listserv and some data harmonization tools there. So as I said, I guess that's my quick overview of what I think is the grand habitat challenge. I have 3 case studies. The first is a paper that came out last year in Progress in Oceanography, and with Lorenzo Ciannelli, who's a professor at OSU, and Mike Litzow, who's now at the Alaska Fisheries Science Center with me. And I'll set it up. I mean, at least a couple of you have heard me talk about this picture before, but this is a picture of Lake Alignagik, which is again in this Bristol Bay river system that supports a large salmon fishery. And you know, I've known researchers at the University of Washington are interested in thinking about how physics affects the thermal environment for fish. So, you know, as climate changes, what are local thermal refuges for fish to deal with climate variability? And so they put out these different thermal, you know, these temperature loggers in the headwaters of this river system and also in the Echo. And the hypothesis is that the headwaters will be subject to melt from the, you know, from these surrounding mountains, and they'll be highly variable depending on the timing of melt. But all of the water mixes in the lake and then it flows out and will be more homogeneous as it flows out. So there's kind of a box model where it's all mixing, and so by the law of averages, the outflow will be more stable temperature environment than the inflows. And that's true in the spring up until a date they found. And at, you know, some date each spring and into summer, you know, some date would occur and then suddenly the outflow would become incredibly variable on this, you know, multi-day frequency. And it would stay that way for the remainder of the summer. And, you know, people thought about it for a bit and eventually realized that what's happening is that, you know, in the summer, the water heats up, it sets up what's called a thermocline. And then the first storm that passes pushes all the water, all the cold water, all the warm water on the surface of the lake to one end of the lake, like pushing water in a bathtub. And then when the storm passes, that warm water relaxes, and that's called seshing. And it sets up a rocking of the thermocline in the lake, like water rocking in a bathtub. And whenever the thermocline's above the outflow, it would dump cold water, and whenever it's below, dump warm water. And so this is one example of many possible examples of how the physical environment sets up a predictable frequency of variability that then is defining sort of the biological landscape for climate adaptation. So we want to do this again, you know, we want to come up with a method of kind of automatically detecting frequencies of variation, you know, major features in the physical environment. And we want to apply this to multivariate, noisy, zero-inflated biological data. So like a dust bunny distribution. And we do that by fitting this package VAST. It's essentially a generalized linear latent variable model, like Dave has written about, and with the catch that it inverts the normal indexing of species and years. So we've, you know, Dave and many others have done work kind of exploring covariance between species at different sites due to factor models. And this is also a factor model. Instead of covariance between species, it's covariance between years. So to talk about this a bit more, I'll use the example of the Pacific Decadal Oscillation. So oceanographers define the PDO as a time series that has a warm or a cold phase, and then a map of changes in surface temperature, sea surface temperature associated with phase. So in a warm phase, there's warmer waters in the Gulf of Alaska and British Columbia, shown in red in that map. And then in a cold phase, it's just the opposite. It's just the inverse of that map. And you know, this is, you know, people have seen this in ecology where during a, I'm going to get mixed up about which way it goes, but the PDO is associated with historical oscillations where Alaska would have good years for salmon returns, and then other times Oregon and Washington would have good years and Alaska would have a bad year. And so there's early work showing that these sort of marine survival of salmon is tied to the PDO or correlated with the PDO. More recently, sort of a cottage industry in thinking about other features of physical variability. So the second mode, the second axis of variability instead of sea surface temperature, it's sea surface height, which is correlated with sea surface temperature, was termed North Pacific Gyre Oscillation, and that's associated with you know, advection along shore and associated upwelling and then associated changes in primary production. And more recently, there's some claims that the NPGO is more correlated with productivity in the Pacific than the PDO. And so this is one example of oceanographers finding modes of variability using empirical orthogonal functions and trying to relate it to community dynamics. I'm sorry, I don't know in the translation to this presentation software, obviously the slide got a bit scrambled. And I don't know how to edit it on the fly, but maybe I'll skip it and just verbally describe. I'll just verbally describe that it's a generalized linear latent variable model in which you have a link function on the left-hand side and then a prediction of the response. And the linear predictor on the right-hand side has an intercept, of a loadings matrix, and a set of maps that are the factors. So here I'm trying to show that I in fact know what I'm talking about by replicating the empirical orthogonal function analysis that's used to define the PDO on the left and the NPGO on the right. So looking at the left, if I use this software fast and apply it to unloaded sea surface temperature for the North Pacific, I can estimate these black vertical lines that represent the index, the association of each year, with the identifying whether each year is a warm or cold phase of the PDO. And then in the bottom is showing the factor loading associated with a warm phase. And so the bottom bright green is a high value, blue is a low value, and that bottom left is supposed to look like, well, it does look like the warm phase shown, in this download off the internet. By contrast, an NPGO is the second mode of variability, but it's the second mode of sea surface height. Sea surface height is associated with temperature because warm water expands and leads to higher sea surface as measured by satellites. But it's not perfectly correlated because it's an integrated measure, not just the sea surface temperature. So it's unsurprising that the correlation is weaker for the second mode of variability, but even then there's a correlation of 0.5, and the map that's estimated by the software is fairly similar to the map, the kind of canonical map associated with the NPGO. So I conclude that we can use this generalized linear latent variable model to generalize how people have previously done empirical orthogonal function analysis. And then the second piece here is that we applied it in the paper to bottom trawl data, so samples of density for 12 bottom-associated fish and crab species in the Eastern Bering Sea, and extract the first 3 modes of variability. I should say that we aren't, unlike oceanographers, we're not demeaning the data, and so the first factor is essentially, in this case, a similar loading in each year, so it's essentially estimating sort of the spatial niche of every species. The second factor is this long-term trend from positive to negative phase, so it's sort of a roughly secular trend in the data. And the third factor goes from these interannual variability to kind of these what are called stanzas, these kind of multi-year oscillations between a positive and a negative. Well, it's, in about 1999, it went to a negative phase, then a positive, then a negative. And I'll skip the variance explained for now. So, the benefit, as Dave has pointed out, the benefit of doing model-based ordination this way is that you can take all the pieces and then reassemble it. So, unlike NMDS or multidimensional scaling or other kind of algorithmic ordination techniques, it's easy to take the constituent estimates and build back what you represent your prediction of the response variable, and that's essentially a joint species distribution model. So in this case, this is the joint species distribution model for 3 species. The bottom row is arrowtooth flounder, so it's a flatfish that has started out primarily in the kind of in this lighter yellow color in the southwest, the kind of slope, the Bering Sea slope, and has progressively moved inshore over time. And I think I'll save the others for later. And then each of these modes of variability is associated with a map for every species. Every species has a separate map representing a species-specific response to this mode of environmental variability. So in this case, each species, the first factor is kind of persistent spatial variation. So arrowtooth flounder, again, in the bottom right of that left-hand side, typically has higher densities offshore in what's called the outer domain. That the second factor goes from a positive phase to a negative phase. And so, for instance, I'll just pull out one example. Tanner crab has a hot spot in this area. This is Bristol Bay. Can people see my mouse here? Maybe give me, somebody give me a smiley face or some other positive seeming emoji if you can see my mouse. Otherwise, I'll just lower hand. Okay. Oh yeah. There's like a pointer. So okay. So here is Bristol Bay, and that hotspot is one example of a species-specific response of that positive to negative phase. So in that case, Tanner crab had a hotspot in this area in the 80s and 90s, and that hotspot went away by the 2000 and 2010s. So that's what that second factor means for Tanner crab. And then the third factor is sort of the interesting ecological story of this system. So in the mid-2000s, there was like a $40 million integrated ecosystem research program that identified that dynamics in this system are driven by the spatial extent of cold near bottom waters called the cold pool. And in this case, that third factor has a 0.87 correlation with cold pool extent. So it's this generalized linear latent variable model fitted only to biology data, and one of the modes of variability it extracts has a 0.875 correlation with physics that's not in the model. And then each of these species has a response to it. So in this case, it's saying that when there's a positive phase of this third factor, which is associated with a large cold pool, a large extent of cold near bottom waters, that pollock has lower density in this northern portion and higher density offshore. Similarly, pollock has lower density in that same area when cold waters are large. Arrowtooth flounder has lower densities kind of south of there. But for instance, this is Tanner crab, has higher densities in that same area. And I think this is Alaska place, has hotspots and density that are totally separate from that. And so these different species are, have interannual variability. It's associated with the same oceanographic driver, but they all have different localized responses to that synchronous environmental variability. So, so the point of this is that we can extract modes of physical variability only by analyzing biological responses. So you can kind of get a shadow index of the physical bottom-up drivers just by by fitting to the resulting biological response. And I guess that's the hook for now. So the paper's in progress in oceanography there, and I'll thank in particular the developers of Template Model Builder, which is the computational engine that underlies VAST, the package. So so a next step that I wanted to explore was trying to think about how we could use EOF but relate it to spatially integrated biological processes. So is there a new way to link spatially distributed physics with spatially integrated biological responses? And that's what I'm calling empirical orthogonal regression, or EOF regression. And I'm presenting results from a paper that's in press as technical advances in global change global change biology. And this is done with co-authors Wei Cheng and Al Herman at the Pacific Marine Environmental Lab. Jim Ciannelli, Mike Litzow, Cecilia O'Leary, and Grant Thompson are all colleagues of mine at the Alaska Center. So again, I'm not sure why this slide got somewhat jarbled, but what it's saying here is that we've got multiple Gaussian processes that have a spatial correlation across space. We then project those with a loadings matrix lambda, L and lambda for loading, to generate covariance across time, and that represents the variability in physics that we're estimating. But the key in EOF regression is that we extract one of the columns of that loadings matrix, and then we use that as a predictor in a separate time series model. So in this case, we're jointly fitting spatially distributed physics using an EOF analysis, and we extract one mode of variability and then put that into a separate time series model. And in this case, that time series model is a linearized Ricker stock recruit model. So a Ricker model is a model for the number of juveniles that are produced per spawning biomass. It's kind of a conventional model for modeling the production of juvenile fish. And because it's easy, because it can be linearized, it's just a nice toy model example for a fisheries crowd. So a schematic showing this, this is the schematic from the paper. So on the left-hand side, we've got the physical submodel, and in this case, we're explaining spatially distributed variation in two physical variables, hypothetically, where each of them has a spatial response to a shared time series trend. So the physics component is inferring this time series trend from the major mode of variability in that dynamics. But then it's jointly taking that time series and fitting it as a predictor variable in a time series model for some spatially integrated biology. And by doing this, it's able to essentially identify the physical conditions, the map of physical conditions that's maximally correlated with residuals in a biological process. So I'm going to show this using bottom temperature measurements, again, in the Bering Sea from 1982 to 2018. Surface temperature measurements at the, winter surface temperature measurements over the same period. And then I'm gonna, the critical piece here is that I'm also gonna bring in an Earth systems model. So it's called a regional ocean modeling system. It's a climate-linked model, deterministic model of ocean physics that is linked to International Panel on Climate Change, IPCC projections of future climate change. And so in this way, I'm gonna use this EOF to intercalibrate historical measurements of physics as well as end-of-century forecasts of physics, and then extract the dominant mode of that and put it into a biology model to see under future ocean conditions how we expect changes in the production of juvenile fish. So this is sort of the plot showing the set of years that are fitted. So in the top left is 1982 through 2016. And then the regional ocean modeling system, we have we have shown here, they're little black dots. It's hard to see, but they become, there's black dots back to 2009. So there's a period of overlap between the Earth systems models and the physical and the field sampling data that allows us to intercalibrate those two different measurement systems. So if you do this EOF analysis on bottom temperature, BT, and sea surface temperature, and compare the observed field observations versus the regional ocean modeling system measurements, you get reasonably good agreement between these different systems. So bottom temperature has a lot of interannual variability. In 1982, 1999 was a cold year, and so cold temperatures in blue got further from the northern Bering Sea down into Bristol Bay. By contrast, by 2090, this ensemble of climate-linked regional ocean modeling system forecast predicts that there'll be considerably warmer temperatures inshore off the coast of Alaska. And by fitting both the kind of historical measurements and the forecast, this empirical orthogonal function analysis is able to extract a single feature across both time periods. It then includes that in this stock recruit model for the production of juveniles, and by doing that, it essentially rotates it so that you can get the map of physical conditions that's maximally associated with stock recruit residuals. And I'll show that in a second, but the effect of that in a second. But I do this separately for 3 different species: cod, pollock, and yellowfin sole. And I guess the key point is that each of these species has a slightly different estimate of the feature that's associated with residuals in the survival of juveniles. So cod and pollock are both expected to have better production of juveniles during cold years. Cold years are associated with cold conditions, cold temperature conditions in this southern middle domain and getting into Bristol Bay. So that's what this map is saying. In years with colder than average temperatures in this area, there's positive residuals in the production of juveniles for cod and also for pollock. But pollock, for whatever reason, seems to cue less on cold temperatures inshore and is more cued on cold temperatures just offshore. So cod and pollock both are generally responding positively to cold years, but specifically have associations with a different landscape of cold temperatures. And then yellowfin kind of has the opposite, where in years with warmer than average temperatures in some of that same area, it has positive residuals in production of juveniles. So if we put this into a stock recruit model, we can compare a stock recruit model without any temperature link in blue here, and we can compare that with a stock recruit model with a temperature link. The top row is for cod, the second row is for pollock, and the third row is for yellowfin. The left-hand side is a time series representation, whereas the right-hand side is this linearized log recruits per spawning biomass. So it's sort of the density-dependent representation of these same data. And the correlation in blue is listing the correlation between the predictions and observations without any environmental link, and the red is with the environmental link. So doing a walled test or using AIC, let's see, I'm gonna get this backwards. I think pollock is significant with, or it's both parsimonious and significant to include the environment. Whereas cod, it's significant but not parsimonious. Anyway. But both pollock and cod forecast under future conditions that there'll be a decline in production of juveniles under future warming. In fact, for cod, it's predicted to be a decrease of in log scale of 0.8, which translates to like a 60% decrease in recruits per spawning biomass by 2090. So, so this again is a new way of taking empirical orthogonal function analysis to extract the dominant one or a couple modes of variability in spatially distributed measurements. But unlike the first talk, this one then uses that as a covariate in a time series model. And by jointly fitting the time series biology and the spatially distributed physics, it identifies the map of physical conditions that's maximally correlated with residual variability in biology. And I think it's useful specifically to generate hypotheses about future climate changes, to also identify the map of environmental conditions that's associated with some biological process. And then that can be used to focus future process research or monitoring studies, where focus monitoring on areas that, locations that are maximally correlated with this spatially distributed biology. So again, this is some of the colleagues from the Alaska Fisheries Science Center, and thanks to many scientists over this multi-decadal sampling program. So finally, the piece I'm currently working on is, how can we put both of these pieces together? So how can we use empirical orthogonal function analysis, or a generalized linear latent variable model that ordinates across time, and how can we fit that to both historical measurements as well as forecasted future climate physics, the physical component of an ecosystem under climate change? How can we do that to essentially produce climate-linked forecasts of ecosystem change? And I think this is an interesting and a non-trivial problem, because a lot of, certainly in the ocean, a lot of the animals we care about are highly mobile. So, forage fish have schooling behavior that is a way of diluting predation. They're also very, somewhat ephemeral, stocks that can kind of ramp up and down population size based on environmental conditions. And then a lot of predators, like in this case, this shark that's shown here, are going to be mobile. Their whole life history strategy is based on being able to integrate kind of chemical chemoreceptors and chemical signals to move physically to exploit patchy prey. And so, if we're going to do climate forecasting for dealing with mobile mobile predators and top-down control, we're going to need to deal with essentially these ecological teleconnections where the predator could respond to environmental conditions that are geographically distant from conditions at a given site. And this is with some of the same co-authors, as well as Lisa Eisner, Dave Kimmel, Louis Barnett, and Kirsten Holzman and John Richard, who also are at the Alaska Center. So I don't know. Yeah, okay. So aside from this piece in here, which I'm not used to seeing in this plot, this is a summary of some of the data that goes into this. So this will be sort of an ecosystem model that includes, we call it an end-to-end model. It includes physics measurements and forecasts. It includes size fractionated primary producers in spring and fall, secondary producers, these copepods, 2 species of copepods in their kind of adult stage. It has a pelagic component, like the sea surface juveniles of age 0 pollock. It has 5 different species of demersal fish and crabs that are caught in a bottom trawl. I'm including data for 2 seabirds, including a shearwater that nests down there in New Zealand, and then also fishing effort that fishes pollock. So there's a protected species, a human component, as well as multiple trophic levels. And then for each of these, I'm trying to keep track of whether it's surface or bottom associated, as well as the malady. So over time, there's the core data is available in summer. There's a lot of data available in the fall about seabirds and also for primary and secondary producers, and so on. So, in a community model like this, you have to keep track of multiple axes when thinking about sort of the spatial and temporal overlap between different data sets. And then this is sort of the same thing, seasonal and vertical availability. So, a lot of, I've got vertically integrated measurements of chlorophyll, secondary producers that are sampled in the spring and the fall. A lot of the fishing, a lot of our survey data for bottom fishes is from the summer as well as bottom temperature. And then it doesn't say here, but I've got fall data for the juvenile fish and for seabirds. So again, I'm applying this empirical orthogonal analysis to all of these variables, and each of them has sort of as I saw, a sort of different annual, and I didn't emphasize, but a different spatial footprint in different years. And so this is the map of average spatial conditions for each variable. So I'll just pull out a few kind of fun little stories here. Right here is I think I don't know exactly how it's pronounced, but Jemchug Canyon. It's the largest underwater canyon in the world. And due to the way that ocean currents flow, relatively warm, deep water gets advected up through that canyon in the spring and brings with it a lot of nutrients. And so this area is the beginning of what's called the Bering Sea Greenbelt, where there's a pulse in productivity due to this upwelling, they call it. And that then attracts squids and attracts a lot of seabirds. And so this primary production is known in the spring to have this higher production near Jemchug Canyon and then Greenbelt. By contrast, large chlorophyll in the fall is elevated here in the Pribilof Islands. The large copepods in the spring have higher density in the south and higher density in the north in the fall, and so on. So you can get these sort of general kind of oceanographic patterns that pop out of a spatial model, and that's no surprise. The model, again, estimates that the dominant mode of variability is highly associated with cold pool extent, so now it's a 0.8 correlation. But because it includes, in this case, decadal scale forecasts, it forecasts that factor into the near future, and it is, basically climate-linked models are saying that future conditions are likely to persist in being warmer than average. And then it also estimates the map of, deviations from climatological average in distribution for every variable associated with warmer than average conditions. So in warm years, Pacific cod moves to the north, pollock moves to the north, arrow-toothed flounder moves to the north. And those are some of the same things we saw in in the first piece of this. But now there's also all these other system components, like, spring chlorophyll has a higher intensity near the Pribilof Islands in warm years. Shearwaters seem to have increased density near Bristol Bay, and so on. And again, it's a species distribution model. So this is it's a climate-linked species distribution model. So it allows us to estimate changes in the distribution of each of these species or these ecosystem components over the next 10 years under likely climate conditions. And for instance, shows that arrow-toothed flounder is expected to continue its movement, spilling onshore. But the hook for this is thinking about community reassembly. And so In this case, you can estimate, you can roll up these species distribution models to look at overlap between different species. So in this case, we're using Schoner's D, which is a measure of overlap. And essentially that in, there was this warm phase here in the early 2000s, then it went to a cold phase for a while, and now we're getting back to a warm, well, we're in a warm phase that's expected to continue. And so in those warm years, we estimate that there's a greater overlap between adult pollock and egg zero pollock or juvenile pollock. There's also higher overlap between arrow-tooth flounder and adult pollock. And so those adult pollock eat their young, they eat juvenile pollock, and they're also eaten by arrow-tooth flounder. And so in warm years, all of these fish that involve adult pollock all move to the same area and have greater spatial overlap. By contrast, in warm years, there's less overlap between fishing effort and shearwaters, and less overlap between the fishery and adult pollock, and some evidence that there's lower overlap between juvenile pollock and their main prey of calanus, of copepods. So again, in general, this sort of ecosystem model that's driven by a climate forecast allows us to do projections of how different system components will change their spatial configuration, and that has been called community reassembly in the literature. So I guess to wrap up, I want to just come back to this grand habitat challenge. So at the risk of putting too fine a point on it, I think the stakes are very obvious worldwide, but the kind of specific hook might vary from place to place. So, I live in Seattle. I guess it was just last year, maybe it was 2 years ago, we had a really terrible fire season, and it was bad enough that there were whole weekends where people were instructed to stay indoors to avoid all of the particles in the air. And of course, it's hard as an American, to watch the fire season that Australia had most recently, which I don't know how that's impacted everybody there. So I think it's a good example of a case where there's ecological teleconnections. So there's physical conditions, regional conditions that prime, drive atmospheric conditions that make fire more likely. There's human development that occurs not just at a given location, but at adjacent locations or geographically distant locations that allow fire to spread across a landscape. There's a lot of successional dynamics, at least in other forest systems, that drive fire. So in the northern, in temperate forests, fires associated with ground cover rates, which is associated with the successional dynamics of forests. And so I think, again, like Australian fires are a good example of all of these different kind of grand challenges of habitat. There's stage structured effects where predicting, fire likelihood and intensity is going to depend on understanding stage structured models occurring spatially. There's non-local effects. There's sort of the need to attribute human development impact both through how it, directly upon fire, but also how it mediates other variables in affecting sort of the resilience to fire. And then there's these counterfactual effects, where, legally we might want to ask what would have happened if we hadn't put an extra gigaton of carbon in the atmosphere. These are all kind of core challenges that I think we face as humans in this coming 100 years. And I think that we, as ecologists and statisticians, need to coordinate our effort to deal with these kind of 4 different topics for habitat science. So with that, I'll put some of the co-authors up again, as well as other people I've discussed with, and I'd be happy to take a few questions.
Thanks, James. That was really good talk. Apologies for the music in the background. I didn't start any CD or anything. There's just a music lesson happening in the background. So I'll try and not speak for too long. Does anyone have any questions for Jim? David?
Hey Jim, thanks for the talk. I guess there's a quick comment. You had a correlation in there between biological signal and physical process of like 0.87. That's that's unheard of for me. Is this a fisheries thing that suddenly you can find things highly correlated with each other? I mean, the ecologists I talk to get excited by an R-squared of like 0.3.
Yeah, no kidding. I mean, that is so much higher than any correlation I'm used to seeing. And I'll say, I mean, I'm doing this research partly to kind of catch up in decades of research that have been done in this kind of new system. I took this new job about a year and a half ago, and it's really satisfying for me to come in and be able to kind of replicate work that was done by my predecessor so clearly. I think the Bering Sea is really unique in having such a clear bottom-up driver of ecosystem dynamics. But I'm willing to believe, you know, for instance, a lot of ice-associated systems like the Ross Sea and the Barents Sea off the coast of, you know, the Ross Sea is down somewhere near New Zealand. You know, I mean, in the Barents Sea up by Norway, and there's a lot of these kind of ice-driven ocean systems that, you know, the extent of ice and cold water is a really strong metabolic driver, and I'd be curious to think about whether there's other kind of analogously strong bottom-up drivers in systems that people work with.
Yeah, right. Yeah, so I had another question on like you're using spatial models across quite large scales. And so whereas often when we're doing spatial modeling, we think of it as points sort of in 2D, like just on a flat surface, it's really sort of curved a fair way around a sphere. So I was wondering if the spatial approach you're using sort of takes that sort of effect into account.
Yeah, so I did build in sort of you know, like a user level switch where you could use polar coordinates to define distances, and I haven't done much with it. I've been more focused recently on trying to build in like barriers, you know, the inlet, you know, a lot of the math that VAST is using is borrowed from INLA, and the matrices are computed by our INLA. And you know, they've done a bunch of work with barriers, and it makes sense in the ocean that you need to kind of, you don't want correlations to go across land necessarily. I guess maybe I'll just say that, you know, there's a bunch of interesting work to do with like non-stationarity in general, like, you know, habitat specific decorrelation rates and estimating, you know, like maybe you have to go further in the deep sea to get to different unmeasured environmental conditions. And, you know, bringing that sort of different mechanisms of non-stationarity into spatial models, I think would be a fun kind of applied stats question. Yeah, thanks again, Dave.
Thanks, Jim. Are there any other comments about James' talk? Seems like everyone's pretty happy and understood quite well your presentation. I would like to thank you, James, for giving a seminar from Seattle at 10 p.m. on a Thursday night. That's very very nice of you. I guess now you deserve a good sleep.
A nightcap and then a sleep. Yeah, thank you for inviting me. Yeah.
And so James' talk will be is recorded and will be made available on the Stats Across Campuses webpage. So if anyone wants to have a look at it again, and then you can also send an email to James if you have other questions.
Yeah. Yeah, thank you.